Okay, there we go. So there's lysozyme and lactoperoxidase. So those are in your mouth. Those help keep the populations at bay. And then, like I said earlier, you tend to see this shift of really high aerobic bacteria to facultatives to anaerobes as the teeth emerge. And definitely there's plenty of biofilm production going on in the mouth, both on the teeth, on the gums, on the tongue. And you have, especially as adults, we tend to have a lot of lactic acid bacteria. But what you eat definitely will influence who's there in your mouth, for sure. What types of foods and liquids are in your mouth will definitely alter who's there because you provide different nutrition for them. And so, and if you haven't thought about tooth anatomy in a while, I'm not asking you to memorize tooth anatomy, but this is a nice visual thinking about the anaerobic pockets that can easily be plenty of surface area when you start to have that gum pressed against the tooth that's emerged. So lots of anaerobic locations in the mouth, even though you know, you certainly bring in oxygen through your mouth on a regular basis, but there's all these nooks and crannies. You have to remember how small microbes are. They can easily find anaerobic sites in the mouth. This is a image of dental plaque looking at the progression over time. So they had these people involved in the study not brush their teeth for 10 days and watched what happened. And on the right are some of the images from that paper. And so this is a slice through the depth of the biofilm and the teeth down here. So you can see, this is greatly enlarged, of course. This is a very powerful microscope image, but you can see that plenty of biofilm, lots and lots of cells in there. So that's why they're saying dental plaque is second to the colon in terms of, you know, especially the density of the microbes showing up per section of the dental plaque. They're even now getting into, okay, what happens when you brush your teeth? And so this is taken from a paper where they did that, and then they tracked who's there, when does the biofilm form, who's in it. And so what they found here is that right, this Streptococcus and actinomyces on the tooth. So that was first in. So those organisms came in first after the tooth. They had them brush teeth and then do rinsing with mouthwash. And so the tooth is right there. Okay, that's the tooth. And then they're showing the biofilm forming. So they were in first. And then the down here, the groups that they found, this Porphyromonas, Streptococcus, and Actinomyces are showing up, growing on the gum surface. And then what came in later, Corynebacterium came in later. Corynebacterium tends to form long, the cells stick end to end to end. So they kind of form a filament-like structure when they're growing. And so this of course is exaggerated. It's not like you have a bunch of things hanging off your tooth that you can easily feel. We're talking really tiny scale here. But what happens is the Corynebacterium kind of anchor one end into the biofilm and then kind of grow off the tooth. And then that provides a surface for other microbes to come in and hang out on. And so again, you have this succession going on of microbes coming in. And then you also have these physical, the morphology of some of these cells really allows for other microbes to capitalize on that and get in and get a foothold. So they're trying to show that there's other microbes here, more coccus shaped, that are hooking on to the Corynebacterium. And, you know, they're showing this is an anoxic zone through that little stretch that they have right here. So they're saying this is anoxic to this point. And then you get aerobic area. So again, the dividing line between aerobic and anoxic can make a huge difference on who's there. So how they interact and how they use oxygen or not can have a really profound influence on who's there and where are they located specifically in the mouth. So they are getting into some interactions where they look at different microbes in the mouth and like, what are they doing? Are they interacting with each other? And what they're finding is that, so some streptococci species, so some of the good ones, some of the bad ones, can be in the mouth area. And then if Candida albicans, which is a yeast, it's a fungus, it can show up too, with certain populations. And what they're finding in this study was that the streptococci were providing a place for Candida to form a physical attachment to. And once Candida physically attached, then it was, living its life, doing its thing, and it was producing lactic acid, when it was going through fermentation. Well, the streptococci like that because they'll use lactate. So the waste product of Candida was being grabbed by the streptococcus for nutrition. And then Candida was also consuming oxygen as part of its physiology. It likes oxygen. And so that reduced the so-called oxygen tension, which created a better environment for the streptococci. They like that. And so this is just one example of things going on in the mouth. And as they watched this progression over time, they saw that these two species formed a nice biofilm with each other and were able to be a little bit more resilient, I guess is the best word. They were more resilient in this interaction with each other when they did the toothbrushing and the mouthwash after that. So they're really starting to get into not only, like who's all there in general, they're starting to look at interactions now too, of what are they doing in terms of their ecology and how might that impact the human host. Yes.
So you're about like thrush in the mouth, is that an unbalance of Candida that's
Yes.
normally there?
Yes, so thrush is when like babies often get thrush, people on certain types of medication will develop thrush because it's usually a certain types of antibiotics because they wipe out the other microbes that can keep Candida in check. And so if Candida gets overgrowth in the mouth, then we start calling it thrush. And yeah, so that is a thing, and that they're definitely looking at that, like, because they've known about that for a while. Like when you take antibiotics, then you can really get this imbalance in the mouth as well as in the gut with the killing so many of the beneficial microbes. So lots going on in the research arena for what's going on in the mouth. Linked to that idea is some species that are found in the mouth. They're now starting to think about, are they somehow part of the process of Alzheimer's disease? Has anyone heard about this at all? So this is a thing that came out a couple of years ago, and it's gaining more interest, that this could really be a factor, a big factor. And so Porphyromonas gingivalis, I would like you to know that one, Porphyromonas gingivalis, because I think you're gonna hear more about this in your life as you go on, because that's the one they're really keying in on for some of the more deep research. But it is involved in gum disease, and when they look at people who have had Alzheimer's and they do post-mortem, autopsies, they were finding that it was in the brain tissue. And then they did some mouse studies because they have a mouse model that kind of imitates Alzheimer's disease progression and Alzheimer's. And they could also find similar things going on when they were doing the tests in that mouse line. So, the idea here is, well, is it the cause of Alzheimer's or just a result of Alzheimer's? And so when this paper first came out, that was unclear. Now there's other papers being published and they're starting to lean toward it could be a cause, not just that, oh, that person got Alzheimer's and they got poor oral hygiene, they kept forgetting to brush their teeth, and therefore you see a lot of Porphyromonas. That was one thought, which I mean, that's legit to think about it that way. But the other idea is, could the presence of Corphomonas in the mouth or brain cause a progression to Alzheimer's? And that's what they're really starting to zoom in on, and it's, some of the data is showing that that could be likely. So again, this idea of good oral hygiene is not just for your mouth, it could be for your brain as well as other parts of your body. You might have heard of some case studies where a person goes and gets dental work done, and then they end up with a heart infection of pericardium, I think it's around the heart. They've been able to document that the microbes, as they're cleaning your teeth or whatever procedure that patient had, blood, they mixed the blood vessels, the microbes got in from the mouth, traveled to the heart region and caused issues with the pericardium. So again, good oral hygiene and taking care without being too, like, don't do mouthwash every hour on the hour. That's not what I'm saying.
That we don't know cause of or cause of bacterial infection. Because like you talk about Alzheimer's, you think of like, we've done a lot of studies with like epilepsy in the stomach, and then I know like endometriosis and things like that, but that's potentially bacterial.
Yeah, we're gonna get into that in part two, the part two slide set. No, it's a great question. And if you talk to a microbiologist who does this kind of thing, they would probably respond by saying, we suspect a good portion of human diseases and metabolic issues are due to an imbalance of the microbes, or certain key microbes are definitely, out of balance. So, but yeah, we're gonna definitely get into that in the next slide set. Did I see another hand? Okay. So now this is Alzheimer's. I also wanted to end on a good note on that. There is another study out that's saying that you might just have microbes in your brain just living there harmlessly that aren't causing any problems. And so this was kind of an accidental discovery. They were working with trying to figure out the schizophrenia issue, and we were looking at differences in brains of people with and without schizophrenia after they had died, and they were doing autopsies. And he said, they just kept coming across rod-shaped objects in all the images that they took, and they were ignoring it because that's not what they were looking for. And then they kind of went, oh, but wait. And so these last two paragraphs, I highlighted a little bit. Then they took 34 samples of different brains, found bacteria in every single brain. The researchers found no sign of inflammation or bacterial disease in the brains they examined. So they were healthy, by all evidence, healthy brains. Most of the microbes were from groups of bacteria that are typically found in the human gut. So here are microbes that normally are in the gut, and yet somehow they're showing up in the brain. So that was an interesting discovery. And of course, they're following up on this, trying to figure out exactly, what's going on there. So more to come on that story. So I'm not saying that if you find, if there is evidence of microbes in the brain, it's always a bad thing. It's certainly something you have to take on a case-by-case basis. So I think all of us can appreciate that the respiratory tract, definitely we have microbes there, especially your upper respiratory tract. The deeper lobes of your lung, you hope, are fairly sterile, but they have documented, even in a non-sick or non-pathogenic state, that there can be microbes there. They're usually not in high population numbers, though. It's pretty low. And we're talking about people who have healthy lungs, not, cystic fibrosis or something like that. So we have, part of our anatomy and what we're just innately born with, we do have a lot of cilia in the respiratory tract that helps with getting rid of debris and microbes. So that's one good way that we have for, so this is the cilia. And so we do see microbes there, but they're usually at low populations in a healthy person. And again, you don't have to memorize who's there unless I specifically tell you to. You don't need to know names of microbes, but I'm just understanding that we can find microbes at low levels in the upper and lower respiratory tract. I like how they have to give it a fancy name, the mucociliary escalator. That's their phrase for the cilia or, sweeping debris up so you can cough it out or swallow it. Mucociliary escalator. That's somebody with too much time on their hands. Come up with that one. All right. Let's go down a little bit. Your genital tract, bladder should be sterile unless you have a urinary tract infection. Urethra, however, is not. All right, so the urethra coming out of the bladder definitely is routinely colonized and usually not by anything that causes a UTI. It's just normal colonization. Certainly, though, you can get players that cause urinary tract infection under certain conditions. And so, of course, those would be the opportunists that maybe you're on antibiotics and your normal flora of microbes that are just normally there are knocked down because of it. That would be an ideal situation for E. coli, for example. E. coli is famous for causing UTIs. There's a couple others that can do that as well. So there's more research now going on, especially when they've been looking at the microbiome of babies versus vaginal birth versus cesarean birth. That has done a lot more now for the research of the actual vaginal habitat. And so they're realizing, so what I'm presenting here is a little bit basic. They're realizing it's more complicated than this with adult in particular. Now with more study, they're realizing that, yeah, some women have lactobacillus species at really high levels, but there's others that don't, and they're both, in either case, they're fine. It's a healthy system. And so you're going to see there's going to be more and more of this. And I think it's being driven by so much of the studies going on with C-section versus vaginal birth. They see differences in pH. So it's not just an oxygen thing that's influencing the microbes. The pH can have a strong influence on that. And different microbes will like slightly acidic versus alkaline. So alkaline environments, we tend to see E. coli is okay with that. And some of the Staphylococci actually can do that. And then, of course, it changes in an adult woman with menstrual cycle. That is definitely an influx of nutrition. So you see a big shift in populations during that time as well. And again, the overgrowth of Candida can happen in the vaginal habitat too, especially with antibiotic therapy going on. And so again, sometimes you have to take antibiotics, but you should be very well informed on what type you're taking. And if you can take a narrow spectrum, it's better to do that if you can, rather than a broad spectrum. Okay, I think I'm going to stop there. The next topic is actually, we're going to get into the gastrointestinal tract, and we're going to spend most of our time there from here on out with a little bit of work into the brain as well. But this is a good place to stop. So we'll get into the gastrointestinal tract on Monday because the exam's on Friday. So, okay. Let me switch into where we're going to start with the exam material. So it's going to be same format. You'll have a multiple choice and then a short answer. And, okay. The first, let me do it this way. Okay, so this slide set, metabolic diversity, part 2, if you scroll down. Whoops. So it'll start with essentially slide 28, at least on my slide set. Slide 28, looking at photosynthesis. Okay. So. That was quite a while ago. Doesn't that feel like a long time ago? So we got to think about that again. So photosynthesis, what I think really the best way to brush up on this is to do a compare and contrast. Like how do plants do it? And that's how I started out with the material. We looked at how plants do it. And especially with the light reactions, we're really kind of focusing in on how do they get their ATP and their NADPH rate. That's really the big thing I'm focusing on for these topics here. And so looking at how plants do it versus and cyanobacteria are going to be very similar. So cyanobacteria and plants pretty much have this system, but then we talked about some microbes only have photosystem 2, some only have photosystem 1, and what's going on there. And so if you did like a side-by-side comparison as you're studying of those different ways of doing it, you'll start to pick out what's unique and what's similar. Okay. So no, you don't have to know all the details of the electron transport chain process. But you gotta know it's an electron transport chain process, right? And thinking about like when cyanobacteria and plants do it, well, they have 2 photosystems. They really ramped up big when they evolved this system. And they have many players in the electron transport chain. But anytime someone says to you, oh yeah, look at this electron transport chain, then you're linking it to, okay, where's the proton gradient? Because there's a proton gradient somewhere. And yes, you are seeing that with how cyanobacteria and plants do it. So you're definitely going to get your ATP, which is great. And you're going to get your NADPH because they're the last electron acceptor at the end. So where are the electrons coming from? Where do they end up? Is ATP made or not? Should be. And then, is it different if they just have one photosystem? What's different about it? So that's how you should approach studying that, because that way you'll be able to pick out similarities and differences. Questions on photosynthesis stuff? Yes.
Or the oxygenic glycolysis that's done by the plants and cyanobacteria. Okay.
Yep. So oxygenic or oxygenic, what does that mean? Produces oxygen. Genic is genesis, to create. Oxygen. So if they're producing oxygen, that's coming from how they strip electrons off of something. What are they stripping electrons off of? Water. That's a little weird how, that's a big deal. Are they using water as their source of electrons or something else? And so you'll see as you compare these, that's a key difference. So if a microbe like cyanobacteria, if they use water, which they do, by definition, they're oxygenic, because as you strip electrons off of water, you get oxygen. Can't help not get it. So water, you're going to get oxygen as a byproduct. They're oxygenic. So water is their source of electrons. But not all microbes do that. They don't want to use water. They evolved other mechanisms. So then they're anoxygenic. So we think a lot about oxygen in the microbial realm. That has a big impact on microbes. And photosynthesis, looking at, what's the pool, what is being used to strip electrons off of it to get this all up and running. Other questions on photosynthesis? Don't forget about the terms down here. So the,
Cycles on this exam, or was that
Pardon?
all covered in the last exam?
yeah, the last exam.
Okay.
We're only doing photosynthesis. Everything else is done. So don't forget about these, terms. You know, those of course would be good, multiple choice question. This one, try to really get this nailed down. You know, this is really a huge concept in the microbial physiology realm. You know, how are they getting their carbon? You know, we're heterotrophs, we eat something that was once living and organic, but many microbes just use it, use CO2, and we're good. And then, so that's their those are your options. In the whole world, planet Earth, those are your options. You're either an autotroph or heterotroph to get carbon, and then phototroph and chemotroph. Well, that's, what are you using for energy? Plants and some microbes, well, they can use light. Unfortunately, we cannot. So we're chemotrophs. We have to ingest chemicals to get our energy needs met. And then the other two huge concepts in the microbial realm is, can they use inorganic compounds as electron sources? We certainly can't do that. But some microbes are funky enough where they're like, yeah, I'll eat rocks. That's cool. I'll get my electrons that way. I'm fine. So lithotroph versus organo. So knowing those pairs, that can follow you out into any other classes you take, in terms of living organisms on this planet. And then the other two slides are just putting them together, these different terms together to describe different microbes in this case. Should we move on? I think the next one was microbial growth, right? So microbial growth. Microbial growth, where are you? You got the shorter version. Okay, so that's looking at some stuff from lab, so you'll feel comfortable with that. Just big picture ideas. I'm not going to ask you specifics of anything you did in lab per se, but you know, understanding street plate, spread plate, viable counts. Definitely you're going to be asked about the growth curve. I told you that before. This is like a classic understanding. Any Gen Micro student should know how microbes grow in the lab in batch culture. And then we just got into some techniques, like how can you work with microbes and count them? Different ways, all have their pros and cons. So think about that. I really like the spectrophotometer and looking at turbidity. That's a really easy way, super convenient. And I can track things happening in real time. But there are some limitations. You got to dilute when it gets so cloudy like this. You couldn't do that. You couldn't take a spec reading of that and get an accurate spec reading. You'd have to dilute that. For sure. And then the lovely membrane filtration process. Direct counting. Questions on any of this? Did anyone go hot tubbing over a break? Oh, so yeah, we got into think about how biofilms form. We're definitely going into that. The endospore, I'm not going to ask you any details of the process. Like, that's just, I'm just trying to show you a nice visual that it's complicated. There's a lot to it. But we're not getting into the details of that. So we already talked about endospores before. I just brought it up again as a way that some microbes differentiate.
under certain conditions. So that was a good example, the heterocysts and the cyanobacteria. And then the funky fruiting bodies that different species of bacteria can use.
Streptomyces with their filamentous and spore production. Yes. So gliding motility, when you think about that, is that on here for this? Where is that? Oh, yeah. So yeah, gliding motility, it's this, you need a solid surface, and so gliding motility gets the microbes moving across a solid surface. They don't do it in liquid, like if they're swimming around in a liquid, they won't do that, but they'll glide around on a surface. And then if you give them like an agar plate that doesn't have a lot of nutrition on it, they'll glide for a while and then they'll realize there's no nutrition here. So then they'll start forming the fruiting body and they'll come together. With the goal of at least have some of these cells survive and produce the so-called spores. That would be a little bit better in some of those other pictures. Other questions on this slide set? What's after microbial growth? Oh yeah, quorum sensing, yes.
I guess with more like the methods and everything, like you're kind of saying, with like the actual like cell counting, we just need to know like kind of like the broad kind of concepts of like what goes on rather than like specific details. Or a little bit.
Yes, and like, no, if there's like a big downside to it, know what the downside is.
Pros and cons.
Yeah, pros and cons of,
And contrasting and comparing between kind of
Yes.
methods as well.
right.
Okay.
Yep, that's a good way to study it. Quorum sensing, so with that, we were quorum sensing nanotube extracellular vesicles. So that, like you don't know who does, like you don't have to memorize which species do which, but knowing like how does a gram positive do it versus how does a gram negative do it, I think that's fair. Again, compare and contrast, like what's similar, what's different. I think that's a great way to study it using that method. Know what the quorum sensing chemicals are in general. So homoserine lactones versus peptides, okay? And that's it. And then like there's the so-called, why do I always blink on that? The one that is, oh, this bugs me. Yeah, but what's the, she uses a phrase, where is it? There, universal microbial language. I don't know why I blanked out on that all the time.
All right, so the AI2, no, you don't, I mean, well, AI2, I guess, is easy enough to memorize, but just know that there are these other chemicals besides homoserine lactones and peptides that are kind of allowing diverse microbes to understand each other. So this universal microbial language idea. So this gets at the idea that you can be from different species or different genera, and you can still understand each other when doing quorum sensing.
What else on this slide set? Other questions? I did ask you to watch that video TED Talk by Dr. Bassler. So that I think would help explain a lot of this too. She's a very engaging presenter. Questions on this? We also did the nanotubes and extracellular vesicles. So that would be too, good to think about, like, what does quorum sensing do for the microbe? What does nanotubes do? What is the extracellular vesicle? Thinking about these different ways that they communicate. Definitely differences, there's pros and cons. We good on this batch? What's the next one? Fungi, thank you. Fungi. Fungi. So 2 slide sets on that. The first one was kind of looking at their biology, overall growth form or morphologies. You need to know the class names. The ones I gave you, there's more, but I only gave you some. So knowing their body form, the hyphae growth, mycelial is with a bunch of hyphae, then we say mycelium. But the hyphal growth form or filamentous growth form versus unicellular, some can do both. Do you remember what it's called if you can do both? Dimorphic, very good.
I'm just gonna ask, should we know each class's life cycle?
No. No, what I'd like you to know is the basic idea that, let's go, I like the Psidiomycetes, where are you? There we are. Knowing the basic idea that if they're kicking into this growth cycle or life cycle, it's their version, basically we're asking, how do they go through sexual reproduction? And so the fruiting body idea, that they've gone through sexual reproduction underground. And this fruiting body, the fruiting body is a mechanism for them to do sexual reproduction. So they fused the plus and minus mating type that fused underground. They built this fruiting body, and then there's this, set area for spore production, sexual spores. So you can have asexual spores or sexual. But I'm not going to get into detail.
Any other questions on, we did 2 slide sets with fungi. So this slide set's got, a lot of just pictures. I'm not going to throw up a picture and say, what is this? Because I didn't tell you, so obviously, no. I just put those there because they're beautiful and I like fungi. So there's a lot in here that you don't have to really worry about. I just love the beauty of it. So, and then the second slide set was looking more at ecology, like what do fungi do in their habitats? How are we using them? Those types of ideas. Viruses are the next topic, I believe. 2 slide sets on that. The basic life cycle of viruses, well, not really life cycle, replication cycle is, I will want you to know that. And then what else did we get into with viruses? Yeah, viral ecology, like what are they doing in their habitats? They're literally everywhere around us. So there'll be mostly multiple choice, short answer, and then the essay portion. So far in the essay, I think I have 3 short essay, and a couple of smaller things, but I'm still tweaking it. I'm guessing most of the points will come from. Well, let me add that up. I can post that later tonight, where I'm thinking most of the points, which topics, I can rank them for you. Let me do that. I'll try to get that done tonight. I'll try to finish the exam.
For the short answer or topic?
Usually topics for short answer are if there's some kind of process. Like maybe with the microbial growth and methods, like how would you look for antibiotic production in a streptomyces or something? Oh, I grow them up in liquid. Use a spectrophotometer, something like that, where there's a process you have to think through, or the viral replication process. Maybe I'd ask something about that. So something that you're like, well, gee, this would be hard to ask a multiple choice question on, because it's more complicated. There's more to it. Those are usually the essay type question. So kind of think like a teacher, could you ask an easy multiple choice on this, or is this a little complicated? Okay, I will be paying attention to email into the evenings from now until, and then in the morning on Friday.